Genetic diseases occur in about 3% of children, right? So if you're an obstetrician, you deliver babies. For every 100 babies you deliver, 3 will have some serious genetic disease. So how will you find out what the genetic condition is? So I'll give you an example. Let's say you have a child who's come in. You deliver a baby. This is your first week as a doctor, and the baby is Caucasian. The baby is blue-eyed, and it does not pass its first stool. Babies are meant to have their first poop very shortly after birth. This has been 3 days, not past its first stool. You think it's genetic, I'm going to put you all up. So what do you do? How do you find out what disease this child has? Yes? Okay, what do we do with the blood? Any ideas? Has anyone got up to this stage at school? For now, any ideas what you could do? Okay, does anyone know what DNA sequencing is? I'm not going to tell you how it happens yet, but just what it is. When you find the sequence of a word, what are you doing? You're finding the letters in their order. So if you're finding the sequence of DNA, what are you doing? Good. Now, how big is a gene, everyone? How many bases is a gene? Approximately in your head. What are you thinking? Any ideas? Pardon? How many letters in a gene? Yes. Pardon? No, just one gene. Any number. About 20,000 plus. So can you imagine finding every single letter of those 20,000 letters for one gene, and then having to check hundreds of genes? How long would that take? A long time. How expensive would that be? Very expensive. So we need faster ways to diagnose children with disease. So what we actually do, everyone, number one, before this child even came in, we've already done huge studies that go by the name genome-wide association studies. What does that mean to you? Break down this big word for me. Genome-wide association studies. Veronica, what does that mean? What is a genome? The genome is all the genes and all the bases in those genes in your body. That is the genome. It's every single letter in your DNA. Does that make sense? Your genome, right? It's the entirety of genes and bases in an individual. And what are we looking for here? Associations. So what I mean by associations, everyone, specifically, is I want you to imagine this is a sequence of DNA, right? And I want you to imagine we've got 100 people. I'm just going to draw 4 here. Let's imagine that 2 of them have a very nasty genetic condition right here. This gene here for them is mutated. And let's imagine 2 of these people are completely healthy in that gene. Now what we do, everyone, is after we've sequenced every single gene for all of these individuals, we start to look around the gene. So just to the right of the gene and just to the... Okay. So we look just to the right and just to the left of the gene, looking for any variations on base that occur in. We look for Snips. And what we notice sometimes is, wait a minute, every time a person seems to have this mutated, I'll do it in purple, every time an individual has a mutated version of a gene, they tend to have, at this Snip location, they tend to have a T. And every time an individual has a normal, healthy gene, they tend to have a G. And when you realize this pattern, this is just pattern recognition, you realize this happens 99% of the time, right? In a population of 20 million people you've sampled. What do you realize now? Do we really have to even sequence every single letter of those 20,000 bases of the gene? Or can we now just start to look for that one letter and look at whether it's a T or a G? Now imagine for this child here, this child comes in, and instead of sequencing that entire gene, you look for one letter, right? That one letter at that SNP location. And what if you find a T? What does that tell you? The child has a very high risk, we can't say for sure because it didn't actually sequence a gene, but has a very high risk of having that disease. Does everyone see that? Because whenever you have a T at that SNP location, what do you also have? The disease gene. So now do you all see how we're using that SNP as a marker for disease? Right? Question?
How do you find the SNP?
Yes, how do you find it? It's a different technique. It's called PCR, polymerase chain reaction, and we're going to talk about that very soon. But for now, I want us to understand the principle as how you use a SNP as a genetic marker. Does that make sense? So SNPs that lie close to a disease gene can be used as a genetic marker for that gene. That is a principle. It's all written here, okay? So right here, you can literally just underline this. SNPs found near a disease gene can act as a genetic marker for that gene. Okay? And this is another formal diagram. If everyone can understand what's happening here, there's a disease gene, there's a healthy gene, and right outside the gene, just right outside here, there's a SNP. And we would have already done many studies on this, but we would have already known that when you have a disease gene, you are more likely to have a thymine instead of a cytosine. And so now, we don't have to sequence the entire disease gene, which takes a long time and is very expensive. What can we do, everyone? We can look for that single SNP. Very good. Does that make sense to everyone? Now, I have a question for all of you. This child, coming back to this child, this example here, this child who still hasn't passed her first stool, who's at risk of getting blood infections again and dying, has a T, has a thymine. Does it 100% mean she has a disease? So how do we know for sure then? We have to sequence after, because then that makes genetic marking useless, right? So how can we increase our certainty in knowing this child has a disease? Yes? Yeah, very good. So correlate with the clinical history. Very good. I like that line of thinking. Anything else with genes? Tips? How many SNPs have we looked at so far? 1. How many SNPs are there in the human genome? Billions, right? They occur 1 in 300 bases. So does everyone agree? If we look to the right-hand side, would we find a SNP nearby? Would we find 20 SNPs nearby? Can't we use 50 SNPs? And then if we find that all 50 SNPs signal that there is a disease gene there, we agree that certainty is pretty much 100%. So in reality, we don't just use one SNP to determine whether someone has a disease or not. We look at 50 to 100 SNPs around that disease gene, and we look at the databases that we have, which has already sequenced hundreds of people's genomes, and we go, all 50 SNPs are associated with the diseased version of the gene. This child must have disease. Does that make sense to everyone? So the way I think of SNPs is they're literally like red or green flags. You just look around the disease gene and you look for whether the flag or the SNP is red, aka it's known to be associated with the disease, or whether the flag or the SNP is green. It's a green flag, Good. Red flag? Worry. Does that make sense? Very good. Any questions so far? No questions at all? Does that make sense now as to why they use the Snips and why they use the genetic markers? Good. All right. Now, we see this word here. Snips can be identified using DNA profiling. Has anyone heard of that word before? DNA profiling. I've introduced two words for genetic technology so far. Sequencing and profiling. Now, a lot of exam questions will ask you to compare the two or evaluate the two. Does anyone know what profiling is? Because you already kind of know what sequencing is. You may not know how it's done, but you know what it is, right? Martin, can you just remind us, what is DNA sequencing? That's it, right? What's the order of the letters, right? It's reading the genome. That is what sequencing is. Now, before we talk about profiling and before we talk about sequencing, I want to just talk about some limitations of Snips. This is not as high yield. It can only come up probably in trial papers. So I wouldn't commit this fully to memory, but just understand. What was the first limitation? I've already mentioned this, right? What was one limitation of just using one Snip? Looking at a Snip is not expensive because it's relative, right? The other option is reading the entire gene. That's looking at 30,000 letters versus one letter. Yeah, very good, right? So the limitations of Snips is that single Snip analysis cannot confirm disease. Multiple are required. What is another limitation of Snips? It's written here, but actually I don't want you all to read that. I want you to just read this word here. If you read the rest of the sentence, you're not doing yourself justice, because this is a really good thought scenario. What can crossing over do to a Snip and its associated disease gene? Think about this. Try not to look at that sentence. I want you to just visualize this. What could crossing over do to the Snip and its associated disease gene? Does everyone agree that we are betting on the fact that that Snip will always be right next to that disease gene? What can crossing over do to that assumption? I want you to imagine a chromosome, right? And you have a Snip right here, and you have the disease gene right here. I want you to imagine crossing over just by chance happen right here. If that happens, what happens between the Snip that is typically meant to be right next to the disease gene and thus inherited together all the time? What happens to that association? Gets unlinked. Because I want you to imagine, what is crossing over? There's another chromosome here, and there's another Snip, and I want you to imagine this is a healthy or good version of the disease, the gene. This is a healthy version of the gene. Now this Snip is signaling that that gene here is diseased, right? This Snip version is signaling or has the base which is associated with the healthy version of the gene. Now if we go and cross them over, what's going to happen? The healthy gene is going to be associated with the diseased Snip. Does everyone agree? What's going to happen is this red flag Snip, or this version, will become red. And this Snip will indicate there is a healthy gene. And in fact, there isn't. Does everyone see that? So in simple terms, the way you write this in an exam is crossing over can unlink a SNP from a diseased gene, reducing the accuracy. That is. Does everyone understand what I meant there? Okay. The diseased version of the SNP can cross over and thus be inherited separate to the diseased gene. Okay, so that is a major limitation of SNPs. Crossing over can unlink a SNP from its diseased gene. And again, that goes back to point one. It reduces the accuracy as genetic markers.
Excuse me, sir.
Yeah?
But don't SNPs just happen like randomly? How would it be used to determine the disease? Because doesn't it just can happen anywhere?
Yeah, you're completely right, but SNPs happen everywhere and all the time. The answer to that question, Lookman, is we already have information from genome-wide association studies. And we have certain patterns that we've picked up, that certain versions of SNPs occur next to a diseased gene. So we use that pattern recognition to screen for diseases, right?
Okay.
So we would already have a database. Who's heard of the Human Genome Project done in 2003? It was like a multi-billion-dollar project where they sequenced millions and millions of people's genomes. That's the first time humans on this planet could read a disease gene and go, cystic fibrosis is a mutation in one letter, right? Or a deletion. It's a deletion in the 508th codon position of that gene. And at that same time, when they had millions of people's genomes in front of their eyes, they could start pattern recognizing that whenever cystic fibrosis occurred, there's a thymine at the 208th position. Whenever a healthy person existed, there was a cytosine at that same position, right? So using that database and then looking at a new person's genome is how we can determine whether they have a diseased SNP or a healthy SNP. Does that make sense, Glenn?
Yeah, thank you.
Good. Okay, now, just a bit of terminology here. Remember I told you we don't just look at one SNP to use as a genetic marker, we use multiple SNPs. The term for multiple SNPs, you'll see this come up a lot, is haplotype. Haplotype, collection of SNPs. Okay. Very good. You'll be given tables with different haplotypes and asked to interpret that information, which I'm going to get to very shortly. It's a very good exam question. Okay. Any questions so far? Okay. Let's get this image here. This is just the methodology for genome-wide association studies. So as you can see, they're getting DNA from diseased people, which are your patients, healthy people, which are your non-patients, and they're looking for all the different variations and bases that can occur. And that's how we identify or already know which letters occur with disease. Okay, good. I'm surprised no one has questions, because I had tons of questions when I learned this the first time. Maybe I'm just not as smart as you guys. Okay, so what I'd like you to do is answer this question here. Question 4. So I wrote this question, and it's a really good question because You do data analysis, and you have to think critically. And I know that students can get intimidated when you see tables and graphs and information that you're not used to seeing. So I want to see how you all cope with that. Can't say anything. It's an exact question. But give it a go, whatever you interpret it as. So that's right, full set, 20 at least, because in about 2 minutes I'm going to stop and ask people to go through their answer. All right. Who's got an answer? All right. All right, let's start with what I would do if I saw this for the first time, which is number one, before even reading everything, trying to answer the question, but understand the graph. What is the graph showing you? Prevalence. Well, what is prevalence? Yeah, how likely, how common something is occurring, right? There's a formal definition you learned in Module 8, because prevalence and incidence are two different things, right? But okay, for now, it's how often something is occurring. Of cytosine at SNP 501, what does that tell you? That is a SNP position. And what are the number of variations you can have at a SNP position? 4. You can have A, T, G, or it's that easy. So we're just looking at how many times or how often in a percentage does C occur between cocaine-addicted individuals and the general population, right? And what do you notice? You see as just, if I ask a kindergarten kid, right, is there a big difference between this red and this red? What do they say? Right? So of course you would say, right, okay, well, the general population has a much higher prevalence cytosine at that base position. Now, what are we looking at here? Are we looking at genes or are we looking at environment? When we're looking at this table, are we looking at genes or the environment? Genes. We're looking at C at Snip 501. That is a genetic region we're looking at, right? So we're noting that heroin or cocaine-addicted individuals are much less likely to have cytosine. Now, let's use our common logic. Heroin and cocaine addiction, those are behaviors that are affected by genes and the environment, right? So typically, it's things like cocaine or addiction are believed to be very environmentally based. But here you see a significant genetic difference between the genetic makeup of addicted users and the healthy population. So what does that tell you about the role of genetics and environment on phenotype, which is addiction in this case? Anyone?
That individuals with less cytosine in probably one or more.
Yeah, which tells you that addiction, especially to illicit substances, is not just entirely environmentally based, but also has a genetic factor. That is the implication of that graph.
Is that a real graph?
Yeah, it's a real graph and real study. Yeah. So that's what I'm getting you guys to actually look and interpret real world data for these questions, right? So the reason this question was hard was because I picked a whole different domain, which is genes plus environment on phenotype, which you understood, but don't really do much questions on it with Snips, right? So basic gist here is you should have realized that there is a big genetic difference between addicted users, healthy population, and so the implication of that is there could be a genetic basis to this underlying phenotype, which is addiction. Yes?
Can you graph like the general population
Addicted to?
cocaine?
So the general population would be everyone who's not addicted, or everyone who does not use, yeah. Sure. Any questions regarding that? Did anyone come close to understanding the graph? Right. Do you now understand that genetics would still?
So how should we answer the actual question about the environment?
Okay, let me break this down for you. 3 marks equals how many points? 3 points, right? So the first thing I'd want to know is whether you can collectively interpret the graph. So mentioning, okay, there is a much higher prevalence of SNP cytosine in the general population. The next mark, where would the next mark come from? It's so important. Yeah, that's 1 mark, but I'm waiting for one other point. Yes, how so? Yes. You don't realize how many times students miss this, right? If you're given a graph or a table and you don't mention the numbers in the graph or the table, you should take away a mark yourself because it's the most basic thing you must be doing. So I would be mentioning there is approximately, what is that? Not a straight line, but about approximately 38% of the population of addicted people have a cytosine versus exactly 80% of the population who are non-addicted. What is the implication of that in terms of the role of genetics versus environment on phenotype. It tells us that since there is a significant difference in the genetic makeup of addicted versus non-addicted people, genes could play a role in addiction. That's it. Does that make sense for you?
Yeah.
Yeah, I'll write it down for you. Therefore, see, the thing is, we can't say genes would play a significant role in phenotype here, but we're just proposing that genes may play a role in phenotype. Okay? Play a role. Because we also agree, everyone, that this could be correlation and not causation. Just because we find that addicted people have a different base letter versus non-addicted people, it doesn't necessarily mean that base difference is causing addiction, right? So remember, causation versus correlation. Does everyone understand this is correlation, right? It's like if I said, people who are Caucasian are more likely to get bitten by sharks. But it could just be that ethnic people in Sydney are studying on a Saturday and Caucasian people are surfing, right? So that's causation versus correlation. Same principle here. Okay? So even though we see a difference in genetic makeup, we can't say that is the reason they're addicted. That could just be some random variation that coincidentally is also occurring. Okay? So good. Does this make sense to everyone? You can ask again if you have any questions. Yeah.
You mentioned how because there's 1 or more % of the population but affected by protein, like it is a relevant.
Yeah, I was going to consider that because that's the first thing I was looking at. Most questions, everyone, the way they'll test you on Snips is they'll show you one variation and another variation, and the variation will be present in more than 99%, which means it's not a Snip. But they've already told you this is a Snip. So you don't need to confirm it's a Snip. If you'd like to, you can, but I'd say you wouldn't get a mark. You could, I'd say, put it in to be safe if you'd like, but it wouldn't be in this marking guideline because they've told you it's a Snip. If they asked you what would you call this variation and why, then you'd say this is a Snip. It's occurring greater than 1% of the population. It's a variation in one letter or nucleotide. Right? Good. Okay. Yes, we're done with Snips, but now we're going to answer Georgina's question, which was, how do we even read a Snip? How do we know whether it's a C, an A, a T, or a G? And how can we actually read entire letters in DNA? Now, I want to stress something really important here. Don't get too pedantic about memorizing every single step of each process. For profiling, yes, you should know this very well, but sequencing has a very complicated method, and they'll never ever ask you in an exam to just dot point by dot point explain the entire method of sequencing. You should just have a general 2-line description of what happens, and you'll be fine for all your exams. Does that make sense? Nonetheless, I'm going to go through the details so you understand it. It doesn't mean you need to remember that going to exams. Could I get... Pardon, could you read out the inquiry question for me? Okay, this is a really vague question. Why should we care about this? Population genetic patterns in a human or any population. When would we look at genetic patterns? Yeah, very good, right? Save lives, that's one. if we can find bad genetic patterns before the disease has a chance to manifest itself, we can treat and cure the condition, right? So one reason we do this is for health, for the diagnosis and early treatment or early management of genetic diseases. Again, a couple of years ago when I was doing pediatrics at Prince of Wales Hospital, a lot of the mums that would come in would have children with Down syndrome or children with what we call, there's a disease called Hirsch Prongs disease. And this is a disease where no neurons or nerve cells go to the bowel. So the child's born with bowel, but it doesn't do any digestion. So you have to surgically remove the entire bowel. And so these children, the rest of their life, have to live with whatever they have, functional bowel, that actually gets connected to the chest wall. And we use a bag that will be where all their food matter will come out into, right? And they often die early as well. So you see a lot of nasty conditions like that, and they're all based in genetic etiology. So if we can diagnose it really early, we might be able to save more bowel, we might be able to manage and increase the life expectancy of Down syndrome. Does anyone know what the life expectancy of Down syndrome is? Yeah, they live till about 68. And that tells you a lot about medicine. The reason they survive so long is, number one, it's not just the medical treatments we give children with Down syndrome, but things like identifying all the factors that are associated with Downs, right? 50% of Down syndrome children will have a heart hole, right? And the fact that we know that means we can go in early, we can screen for it, and the second we pick it up, surgically fix it up. They live longer, right? So things like that in medicine can prolong and change lives. Now, why else should we care outside of health? Two are the main points the syllabus wants you to consider. Yes? Yeah, very good. It can. This is another point. It does link to the syllabus. I'm going to put it here. I'm going to put it in black because the syllabus hasn't mentioned it, but it's a very important point, which is when you find genetic patterns, you might be able to treat them with genetic technologies. This is a bit of treatment, genetic technologies. This is already happening, by the way. It's currently illegal to go in and change the genes for a spermatozoa or an egg, because if you stuff up that egg and all of its lineage will have that step up or mutation, right? But someone in China did that, and they cured 3 babies of, I think it was HIV, or they had some other incurable disease. So the doctor actually went in and germline edited that gene out, which was questionable because they did something illegal. They saved lives. So this is actually happening right now, which is exciting because this is a new revolution in society and humanity. Good. Anything else? Think outside of humans, right? We're not the center of the planet. Any ideas? Conserving other species, right? So if we can identify the variation in other species, we can identify organisms that are at risk of going endangered or extinct, and then put conservation programs or protocols in place. Anything else? One last one. This is probably the least important, but it's very interesting. Where did we come from genetically? Yeah, you could say that the lineage of evolution, life began on water according to science, right? But let's take a few steps forward. Which continent does science propose we? Why? Why is that? So, does anyone know why we think that all humans originated from that continent? you should question it, right? You can't just say, oh yeah, science is right all the time. So I'm not going to answer that question until we get there. But human evolution. In fact, genetic technology has answered the question as to where we come from. We'll also talk about that. Good. Let's start with the first dot point. So, Prithvi, could you read that one out for us?
Investigate the use of technologies to determine.
Very good. Now, we're going to start with DNA profiling. Now, one thing I want you to highlight next to profiling is determining the genotype. That's all you need to know. Determining genotype. What is genotype? Genotype. Yes? Sorry, could you repeat? Yeah, genetic makeup, very good, right? So, very good. Phenotype is a physical makeup of an organism, so genotype is going to be the genetic makeup, or genetic makeup. So, genes and alleles. So, identifying genes plus alleles. Very good. Now, it's often called, in a colloquial term, DNA fingerprint testing. Now, I want you to remember this. This actually has nothing to do with your fingerprint itself. What's unique about human fingerprints? Yes, the unique thing is that they're all unique, right? DNA fingerprint testing, it indicates the same principle, that we are looking at the DNA to identify unique variations between people. We're not looking at the fingerprints at all. It's called DNA fingerprint analysis because they're looking for unique variations in those genes and alleles. Does that make sense? Okay, it's a colloquial term, and fingerprint testing is the exact same as profiling. Okay, good. We don't actually look at the DNA in the fingerprint. Good. Now, I think we've hit the hour mark, so before I overload you with what profiling and how it occurs, I think you ought to all take a 5-minute break. So stand up, stretch, feel free to go outside, get some water, and when we come back, we'll touch upon PCR, profiling, and then sequencing. And guys, I think after that, maybe one more lesson, and we'll finish the module. Any questions? Kirtana, Prithvi, Lukman, you had a question?
With the question, the graph question above,
Yeah.
you said that there is significant difference in genotype of individuals that are addicted and not, but wouldn't it be because of environment that resulted in different genotype?
You could make that point, but remember again, where are Snips more commonly found?
Non-coding DNA.
Good. So would it be expressed in the active?
Oh, yeah, right. Yeah.
So
Okay.
that's an assumption you have to make. You could mention that point, Lukman, if the SNP was located in the environment, or if it was located in coding DNA, it would be acted on by natural selection, and that could account for the prevalence or difference. But degree, you're assuming the SNP is in coding DNA, which is actually not very common at all.
Okay, so the main implication is that genes could play a role in people who are addicted to cocaine.
That's
Okay.
all you have to mention. Genes could play a role because there is some genetic difference between addicted and non-addicted people.
Yep.
Good question. Raya, any questions at all? And Kirtana, Kushi, any questions?
No.
Good, thanks, Kushi. Yeah.
Didn't we mention the Huntington's disease question last week?
Which disease question?
The Huntington's disease is like people.
I don't think did we go through Huntington's? Yeah, we might show you the question, so I remember. This one, right? So, yeah, so the hard part of this question is just information, right? So the first thing that started is first one, malaria is a disease caused by what? Not just mosquitoes, but mosquitoes that transmit the protozoa, which is the bug. Okay. So that causes diarrhea and death. Then there's also another condition called sickle cell, in which people are protected from bug or malaria. Now, where is malaria more common, in Africa or Europe? Europe is so. This is why you make a little thought experiments. Maybe get a pencil and be right here. And did you need on ACE as well? What's going to be what? That's good. Okay, so you talk about oh wow, okay. Okay, so there is malaria, so much right Africa, then Europe. Which region has malaria? Good. So shaded around malaria. Now, there's sickle cell disease in both these countries, right? In which country is it favorable trait to have sickle cell? Good. I want you to write sickle cell disease in Africa. Because if it's favorable, what's going to happen to people that survive? They're going to reproduce, pass it on, more and more people will get sickle cell trait. It's going to keep them going on. Those people have sickle cell trait in Europe, what's going to happen to them? Yeah, it's going to be constant, it's going to go down because it's not favorable. There's no malaria in Europe. Then what's going to happen to the sickle cell disease in Africa? Which country does it go up? Africa, because it's being acted on by that natural selective pressure in there, where it's a favorable trait. So in each country, sickle cell disease favorable traits under selective pressure. No selective pressure, not favorable. Yeah. Yeah. Yeah, I do. Like, you do a lot of research. I like this too. So, you know, doctors, they don't know everything. So, we actually have databases, information on all the treatments that you guys can't have access, right? I can screenshot that and send it to you. Use it. Yeah, so you talk about countries, right? We can do right now. What is it cost? Information process. Show me a marking criteria because that the way you do this assessment is have a mark criteria out into every single point. Yeah, send me the marking criteria. And also message me on Teams because I'll probably send it to you after the class. But did you have any questions about 1A? So I think we'll get to 1A. Do you have any questions at all about 5? I know you kind of started with the bed. Did you look at 5? The early stuff at 5?
I don't know what schools and stuff, but I think I would do it.
Yeah, the thing is, you don't need to know the earliest stuff to learn this stuff. It's all new stuff, all the students as well. So, I wouldn't regret that. Have you used linear replication before? So, like, LKs and enzymes as well, very good. So, you know that, you'll understand all of this as well. I think All right, let's continue. So everyone refreshed, ready to get into this? Because this is a bit harder conceptually for Mod 5. Now, we'll start with profiling all the way down here into how it occurs. And to understand how it occurs, I'm actually going to ask you all a bunch of questions. And from those questions, you'll understand how it occurs. So Ethan, starting with you, when does DNA replication occur? DNA replication. And the other M. Mitosis. Happens in both, right? Which phase, Pranav? Good. Which part of interphase? Because you have to remember the cell cycle looks like this, right? Mitosis, that is mitosis. That tiny step there, right? There are 3 phases before that, which is the majority of the time that the cell stays in, right? And then this will be a cell after it leaves the cell replication process. There's a G, the S, there's a G phase. Does anyone understand what's going on here? Want to give it a go? The SG, just read it. S happens there, right? There's a G0, there's a G1 phase, and then there's mitosis. You don't need to know the G phases, right? G stands for growth, okay? And the way you understand this is mitosis is not a cell division, it is a nuclear division. The goal of mitosis is to only divide the nucleus. It doesn't care about the other organelles. The other organelles actually divide here, where organelle division happens. The endoplasmic reticulum, all those organelles divide there. In this phase here, the S phase, remember, this entire thing here is interphase. All of that is interphase. The rest of it is the PMAT, right? Do you see the scale of things occurring, right? So in the S phase, the S stands for synthesis. Why? Because you are synthesizing DNA. Now the reason I'm bringing this up is this technology, PCR. It was derived from just understanding what happens in DNA replication, then artificially replicating that. Okay? So, does anyone know where we use PCR nowadays? Businesses are making so much money. To actually come back from the US, I had to pay $400 USD to get my nose swabbed, right? That's like 600 AUD. That's like a trip to Bali, right? These companies are minting money off this process here, which is PCR. Now, the goal of PCR is to amplify DNA, right? Because if I had COVID, my nose would have had some COVID RNA. But it's very, very tiny amounts because there's not much virus in the nasal orifices. So what they do is they take the swab, that small COVID RNA, they'll add all the enzymes and ingredients required to multiply that until it's so large in terms of the number of RNA strands that it's detectable on a huge machine. That is the goal of PCR. Okay? So here, what we're going to try doing is we're going to look at DNA replication and then create profiling right now. So what's the first step? So now you understand this is an S phase of interface. That's what I was looking for. How do I make this DNA replicate? What's the process? What's the first step? Unzip. So what enzyme does that? Yeah, helicase, right? So helicase does that. Unzips and unwinds the double helix. What's the next step? Nisha. Yeah, but how does it start to replicate? Did you say P5? 3' to 5'. Yeah. Yeah, so the first next step is a primer, or an RNA primer needs to attach. Why is that? Sorry, a bit louder. Very good, right? The primer is kind of like if you're zipping up your jacket, it's actually a zip, right? For that zip to move up, it needs to actually have an attachment point to start. The same way, that's what the RNA primer does. It serves as a starting point for polymerase to bind and then start propagating. Very good. So the RNA primer binds, and then what's the next step? Next step. Yeah, right, because this is all occurring in the nucleus, which has letters. It has nucleotides free-flowing. So polymerase will read an A, will attach a thymine, right? Good, so it keeps doing that. And then finally, Georgina, what's the final step? So right now what we've done is we've built new strands. These are the 2 new strands. What's the final step? It's all logical. Actually, you don't need to memorize much. If you just build new things, if you build a new wall, you put bricks to do you need to put between them to reinforce it? Cement. So what is a biochemical cement of the enzyme that produces that? Yeah, very good. So finally, the final step is ligase. Now, we are going to replicate this, right? So I want you to imagine you're in your little science workspace, and you're going to create the technology which companies are making billions of money off right now. So the first step is we need to tear the DNA apart, live and agree, right? Now, instead of helicase, because helicase is quite expensive, we can just use high temperatures. Because what bonds exist between base pairs? Hydrogen bonds, right? And any of the online students, do you know what temperature is required to break the hydrogen bonds of DNA? What temperature?
72.
Oh, very, very close. I know why you said 72. 95. You close enough, I'll give it to you. In an exam, you probably get that. Good, very good. 95 degrees. So instead of helicase, I'm going to write in green now what changes we're making. Okay? So instead of helicase, we use 95 degrees to break the hydrogen bonds between bases. Good. Now, what's the next step? We need RNA primers, right? So we did the same thing. So just like in natural DNA replication, in our PCR, the next step is we're going to be using RNA primers. Now, RNA primers are quite temperature specific because you'll have to understand what temperature does DNA replication occur at? Normal DNA replication, what temperature does it occur at? Yeah, about 36.5, your body temperature. But what have we just done, everyone? We've brought everything up to 95 degrees. That's going to fry every single enzyme in the human body, right? You would have learned in year 11, enzymes can denature or get destroyed at very high temperatures, right? So we use a special RNA primer that can withstand the high temperature, but we also make sure that we can change the temperature. Remember, all this is happening in a machine. I'll show you what the machine looks like. This is the machine which is doing, it's called a thermal cycler. And what you do is you put a little, see this little test tube here? You put that little test tube in the thermal cycler, that little test tube has a sample of DNA or RNA, and the thermal cycler will do the rest of the work, okay? So we have the added advantage that we can start moving temperatures around. The best temperature for RNA primers is 55 degrees. This thermal cycler will initially go to 95 degrees to rip that double strand into single strands, and then it will go to 55 degrees so the primers can bind. Does that make sense to everyone? Now, there's a fancy word for primers binding. It's called annealing. Has anyone heard of that before? So the technical word we say is RNA primers anneal. Anneal means attach or bind. Okay? Good. Now, the third step is, we've already mentioned, polymerase, right? After the primer, polymerase needs to bind and start acting. Now, if we were to use a human DNA polymerase, what's the issue here? Go back to step one. What do we do that would have destroyed any human enzyme? 95 degrees. 40 degrees kills a human. If you have a prolonged fever for 40 degrees, you die because all your enzymes denature, your chemical reactions stop, organs fail, you're gone, right? So 95 degrees, that's just frying enzymes instantly. So we can't use a human DNA polymerase. So scientists did all the research experiments, and they found an amphibian DNA polymerase that can withstand 95 degrees. And lo and behold, the name of this polymerase is called Taq polymerase. And the cool thing about it is that it operates best at the temperature Luckman mentioned. What temperature did he say? 72 degrees is a temperature which Taq polymerase operates best at. So now this thermal cycler was initially 95 degrees to rip the DNA in half, went to 55 degrees to cool and allow those primers to bind, and now it's going to go back up to 72. You need to memorize these temperature changes because I'll show you on a graph that I'll ask you to explain what's happening, etc. Okay? 72 is for the polymerase to start working. Very good. And it will just repeat. Those are the temperature changes in PCR. Right? So I guess my next question is, what needs to be in the PCR tube? Because the thermal cycle is just going to go 95, 55, 72 repeats. It's going to keep doing that. And every time it repeats, it doubles the amount of DNA in that sample. Right? Because what you will notice, in that one round, we went from 1 strand to 2. And if we do that again, how many strands do we end up with? We do it again, how many strands? Do you see how it's exponentially increasing? If you do this for 1,000 rounds, can you imagine how many strands we would have? I think billions, because it's an exponential, just doubling, 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 right? So now I want you all to answer the question, what needs to be inside the test tube? So let's say you swapped, so you own one of those PCR companies, and you're a multi-billionaire. What do you need to have in your test tubes? Your company, what needs to be in the test tubes? You need to source those raw ingredients, right? So you need to have a solution for everything to be contained in, right? What else needs to be in there? Yeah, very good. So we need to have the DNA sample. What else needs to be there? Yes. Yeah, what enzymes? Do we need helicase there? No, very good. Skip it. We're going to use a thermal cycle. So what do we need after that? Good. Primers aren't an enzyme. Remember, they're just RNA strands, but you're right. We do need the primers, RNA primers. What else do we need? Very good. Taq polymerase. Typically, no, you actually don't. Because remember, the goal here is not stability. We don't care for all the DNA to be stable because it's not going to exist in an organism, right? This is just amplify enough so we can say, the virus is there, right? We don't need a ligase. Very good question. Anything else? I'd say that's pretty much everything. And then the solution, as we mentioned. It's often called a buffer solution. For those of you doing chemistry, you know exactly what a buffer solution is. Now, do you want to explain in the most simple way? This is going to test your skills as a teacher. What is a buffer? Have you learned buffers? Have I taught it to you? Oh, you got the hook. Very good. Nailed it. Solution where the pH will not change. Because think about this. Enzymes hate temperature changes. What do they also hate? So we need a buffer solution so the pH does not change while all of this is happening. Okay? Very good. Now, let me tell you what they'll ask you in an exam. They'll show you a graph of the temperature changes, ask you to justify why that's happening. They may ask you what's in a PCR tube, or they may ask you to just outline the method. So as long as you know these general steps, you're good. Okay? Now, I still haven't answered your question. I've turned one strand of DNA into billions. What, do we just taste it? How do we know that there's billions of strands, right? Does anyone know the answer to that? How can we tell that there are billions of strands that are positive? You are very late. What was the delay just now? Okay, you need to message us or let us know, okay? Because you missed out on more than half of the class. So you need to watch the recording as well, okay? Okay. All right, so so with PCR, you need to know the general steps. And so the question is, how are you going to convert that or visualize the PCR result? Yeah, the DNA, but you still won't see it. It'll still be a solution, clear solution.
Yusuf?
Yeah?
Could I ask that question?
Yeah.
Yeah, gel electrophoresis.
Has anyone heard of that word before? Gel electrophoresis. Very fancy word. Look, man, I want you to give it a go. What is gel electrophoresis?
So it's basically just a way of visualizing each amplified allele, so STR or Snips, and each of them identifies a band. And it's
Very
just a
interesting.
way of, you could use it, to, yeah, that's pretty much it, I guess.
Okay, I like that. I understand that you understand it. Now, electrophoresis is like the yang to PCR, if that's the yin, right? They're literally complementary. You can't do one without the other. If you just PCR, you can't tell anything, right? You need to do electrophoresis after that. Electrophoresis is a way of visualizing DNA. That is all I want you to understand for now. And I'll show you how we visualize this DNA. So this is what an electrophoresis vat looks like. Okay? Now this is your amplified PCR sample. Amplified PCR sample. Okay? And so what you do is you will pour this PCR sample into this apparatus. Now what you're seeing here, everyone, is all of this is jelly. It's agarose gel. Agarose means it comes from seaweed, right? It's a clear gel. And it's been built in such a way that there are depressions. You see these little slits here in the gel? Literally as if somebody poked a long rectangular hole, so it goes all the way down to the bottom of that gel in intervals, as you see there. Okay? And what we do is we use a pipette and we transfer the solution into the well. Now I'm going to ask you a question. DNA, does it have a charge? Specifically the chemistry students. Who does chemistry? Good. What is the charge of DNA? Any ideas? This is where you look at its structure, you're transferring that. And does anyone know?
Yeah.
Anyone other than Lookman? I know Lookman knows electrophoresis. Why? Can you explain to the class why? That's right. Exactly the case, right? So the sugar is neutral. The nitrogenous base is more or less neutral. The thing that is charged about DNA is phosphate. Phosphate has this chemical formula. You don't need to know. I'm just showing you so you understand this. That's 3-, or strongly negative. Okay? And as a result, DNA entirely is negative. So what happens if you put a positive charge near a negative charge, everyone? And what if you put two negative charges next to each other? What do they do? So what we're actually running here, in very simpleton terms, is we're racing DNA. We're racing our samples, right? So this pipetted sample is going to have thousands of strands of that DNA because you've replicated. Does everyone agree? Now, as soon as we turn on our power supply, at this end, there'll be a strong negative charge. What is that going to do to the negatively charged DNA that's sitting in this well? It's going to start running down. But we've replicated it to thousands of strands. Is this a skinny runner or a fat runner? It's a fat piece of DNA. It's humongous. So will it go through the gel fast or will it go through the gel slow? Very slow. What will happen is it will be so slow that it won't even finish the race. It'll probably go till about here, then it will stop. Okay? Now, I want you to imagine that we use someone else's sample, right? Someone who doesn't have COVID in this case. So we'd repeat this PCR process, but this person does not have COVID. Now, the primers, everyone, only bind to COVID DNA sequences. Remember, the primary seq is unique to the sequence of DNA being analyzed. The RNA primers we put in are specific to the COVID RNA. So if you don't have COVID, will the primers bind to anything at all? Will polymerase replicate anything? So even despite PCR, we'll still only have maybe 1 or 2 strands. So this healthy person, when we pipette their DNA in here, right, their DNA is only 1 strand. And so when you turn on that negative charge, what's going to happen? That single strand will be repelled, but will it move further or will it stop earlier? Remember, this is a skinny runner now. It's going to run way further, all the way down here. See this bend. Okay? And then the final thing we do, everyone, is we get a known COVID sample. This is called a control, right? We get a sample that we know has COVID, and we run it through it, and we see, okay, the sample that has COVID has stopped right here. Now I want you to imagine a bird's eye view of all this. What is this going to look like? It's going to look like, let's draw it together. It's going to look like this. The vat will look like this. There'll be your wells, right? These wells here. There will be your positive control, which is here. There will be patient sample 1 that is here, and patient sample 2 that is here. This is your positive control. This is patient 2. This is patient 1. Now you're at the lab. You see this. How do you interpret this? Georgina? What does patient 1 have? Yeah, right? Because their band has aligned with the positive control, which we know is COVID. It's that easy. What about patient 2? The sample's gone all the way down, which indicates that nothing replicated, okay? And so they're negative, right? So this is how we use PCR to diagnose infectious diseases and also genetic diseases. Because think about this. This could be the same case for a genetic condition. We could get a known sample of, pick a disease, Martin, pick a genetic disease, your assessment task, what is it on? Huntington's disease, right? Huntington's disease is scary because if either one of your parents has it, you are definitely getting it, and you get it at about age 60, you can't cure it and you'll die. So imagine you're a 20-year-old, your mom, your father had Huntington's, you are doing the test, finding out whether you have it as well, right? Now, this is a positive control for Huntington's disease. This is a known sample of Huntington's. We PCR'd it, it stopped right there. These are the 2 patients. Who has Huntington's? Patient 1. As everyone see, their sample is aligned with that positive control, which means they have the exact same sequence. Patient 2, nothing replicated, right? They had the healthy gene. The primers could never bind. And so their tiny DNA sample moved much further down the well. That's a negative. So do you see how we can use PCR and electrophoresis for the identification of genetic diseases? And number 2, also for infectious diseases, especially ones that have like DNA, so viral DNA. So this is mainly for viral diseases. We'll do PCR. Yes?
How do the rapid tests work?
The rapid antigens, that's more physics than biology. They actually work on fluid mechanics for diseases versus non-disease. So you can actually search the name of the technology. It's called lateral flow assay techniques. That's more chemistry and physics. And that's where you start to realize biology, chemistry, physics are all just different parts of the same coin. So when you delve too deep into biology, you go into the chemistry of things. You delve even deeper, you go to the atoms in physics. So if I answer that, it'll take a whole other lesson. Good. Any other questions?
Yeah, excuse me, sir.
Yes.
What makes the DNA heavier or lighter?
Yeah, very good question. I'm going to answer that very shortly. Does anyone have any other questions? Yeah.
What if the patient wanted
Yeah, so people have different, no, you wouldn't retest because people have different genes. You can have a healthy version of a gene that's just different, right? So anything that doesn't align with a positive control is a negative result, okay? I'm going to answer your question very shortly. But in the meantime, I want you all to read these notes too, because what you've done so far is you understand the concept, but I can guarantee none of you can write this in an exam yet. So I'm going to teach you now how to articulate what we just learned in an exam question. How do I write a 2-mark on PCR? How do I write a 2-mark explaining electrophoresis? What do you need to know about gel? Sorry. Yes, that's what I'm going to explain to you all now. So now you understand the conceptual side of things. And Lukman's question was, do you repeat your question, Lukman? I'll answer that now.
What makes the DNA heavier or lighter?
Yeah, what makes the DNA heavier or lighter is solely dependent, Lukman, on what DNA sequence is present, right? So let's use the COVID sample as our example. So in patient 1, who is positive, and we'll get patient 2 that is negative. In patient 2 that's positive, Lukman, would our RNA primers anneal to the sequence? Remember, we always select specific RNA primers to whatever gene we're trying to amplify. If I'm trying to amplify COVID and this patient is positive, will the primers bind? The primers will bind. So will the replication occur? Will it occur multiple times? Exactly. So this one strand is going to amplify into multiple. And that's going to create a very heavy DNA fragment. Does that make sense, Lukman? We say it's heavy because instead of one strand, there's millions of strands.
Yeah, okay.
But in a patient who's negative, would the RNA primer anneal?
No.
No. So there's no annealing. And so instead of one strand going to multiple, we only have one strand in the sample at all times. And so we call this a light fragment of DNA.
With PCR test like happen?
Literally this way. This is the way that
What about I guess fake positives?
Yeah, so false positives happen when primers can accidentally bind and anneal to correct sequences as well. The false positive rate with PCRs is quite low. That's why if you're positive on a PCR, you're probably positive. False negatives can occur, right? That's more common, where even with the COVID sequence, just because the primers don't float around in solution to the actual sequence, they don't multiply enough, and so they don't stop at the appropriate position. That can sometimes happen. But false positives are very rare because the primers are very specific to whatever we are trying to add. Okay. All right. Okay, so guys, before I show you how to explain electrophoresis and PCR, what I've actually got is one of my ex-students is actually going to come in and quiz you / teach you a little bit of protein synthesis. So I want to see how well you guys know the process. So it's going to be very discursive, I guess. And I want to see how you go. So don't disappoint me, okay? Martin, you're probably the only one off the hook because you this time, okay? Okay, so what I might do first is I will set
Hey everyone, nice to meet you. So yeah, as Prithvi told you, I'm one of his ex-students and I did biology as well last year. And I'll just give a quick introduction to myself. So I graduated from Wilson Agricultural High School last year and I'm doing medicine this year at Western Sydney University. So yeah, I take an interest in biology and medicine. So yeah, so we'll just get right into the lesson. So the reason we're doing protein synthesis today is
One
because...
thing, I want you to pick on that.
Okay, I'll pick on that.
These guys,
Alright.
not off the hook.
So
That's the point.
so the reason we're doing protein synthesis today and revising it is because it's one of the most important concepts in HSC biology and it always comes up in your exam. So Prithvi's told me he's already taught you protein synthesis, so I'm going to be testing everyone, okay? So I'm just going to randomly pick on you and you have to answer me, alright? So protein synthesis, also known as polypeptide synthesis. Okay. Okay, so we'll just start off with the basics. What is protein synthesis? Can you tell me? Yeah, so it's, in simple words, just the process of making proteins. Now here's a hard question. What are proteins? Yeah, but what do they do? What is their structure? Like, why do we need them? Yes, that's true. They can determine phenotypes of organisms. You've heard about proteins when you're working out and people tell you to eat proteins and they'll help you grow muscle. So another structure and function of them is the development of muscles. And there's hundreds and thousands of different use cases of proteins, which is why they're so important. So in short, proteins, without them, you can't function, you can't survive, which is why this process is so important. So what are the two main stages of protein synthesis? Transcription and translation. Okay, so which one comes first? Yeah, I just remember it because they both start with trans and transcription has a C, which comes before an L, so that's just how I remember it. So, okay, so we all know it starts off with a DNA strand, yes? So, why does it start off with a DNA strand? What's exactly special about DNA that allows us to make proteins? Yeah. Yeah. So you need genes to make proteins. And we all know what genes are, right? Genes are just, you've heard about genes. Someone might have genes for black hair, someone might have genes for brown hair. But just for the sake of protein synthesis, we can say that genes are the instructions to make proteins. So you need genes to start off the process. So you're going to start off at a gene, okay? So we'll just pretend that this is a gene here. This is a DNA double helix structure. So what's the first thing to start off the process? Can someone tell me the first thing we need to start off the process? Could you tell me? Sorry? Yeah, RNA polymerase. So RNA polymerase is the first enzyme that you need to start the process. And where does it attach? Well, it attaches at something known as a promoter sequence. So the promoter sequence, all it does is it tells the RNA polymerase where to attach. So the promoter sequence is obviously going to attach on a gene, yeah? That's where it's going to start the whole process. And RNA polymerase is going to bind on that DNA sequence at the promoter. And what is it going to code? mRNA strand. Okay, now let's just quickly look at what an mRNA strand is. So can someone tell me the difference between DNA and RNA? Single-stranded? Yeah, those are the two main differences that you need to know. So it's single-stranded, and it uses a U instead of a T. All right. So we know that the RNA polymerase moves along this DNA strand, and it codes an mRNA as it goes along. So this is something that a lot of students get mixed up on. It was in our task 2 at Holston, and everyone was just mixed up about this. So what are the name of these two different strands?
Bottom one would be the coding strand.
Okay, so you think, so you're saying the top one should be the template strand and the bottom one should be the coding strand?
Oh wait, never mind, I'm looking at the wrong arrow. So top one is coding strand.
Yeah. Yeah, this is just something small to remember. So obviously the bottom one is going to be the template on which we base our mRNA strand off, and the top one is just called coding because it's the same as the mRNA strand, but it's going to use a uracil instead of a thymine. So yeah, so that's basically transcription. It's just going to be an RNA polymerase coding for an mRNA strand, and once it's done, are we finished with transcription? What do we need to do? Splicing, yeah, splicing. Does anyone know the name of the enzyme we use for splicing? Yeah, spliceosome. This also came up in our exam, and no one knew what spliceosome was. So, yeah, so to recall, if anyone forgot what splicing is, we have our gene here, yeah, we have our DNA here, and we've got an mRNA strand which is based off this DNA. But I'm not sure if Prithvi has told you already, but only 1% of our whole DNA structure is actually made up of coding DNA, or genes, right? And 99% is actually non-coding DNA, so it's not going to actually code for the proteins. So it's just going to be non-coding. So throughout this whole DNA strand we have here, there's going to be some sections which are non-coding, and we're going to get those in our mRNA strand here. So we want to get rid of those sections, and we only want to keep the coding sections which will code for our proteins. So does anyone know the names of the non-coding and the coding sections in this strand? Which ones are introns? Yeah, so we'll just pretend these are the non-coding and these are the exons, yeah? Okay, so using spliceosome, we're going to get rid of the introns and we will end up with a mature mRNA strand, which is only made up of exons. And there's a final step which is done in splicing. Does anyone know? Yeah, poly A tail and? Yeah, a 5' cap or a met cap. And this is on the 3' end. And does anyone know why we use these caps on the end of this mRNA strand? Yeah, so it doesn't degenerate. So I forgot to mention at the beginning, all of this is happening inside a cell nucleus, yeah? Because that's where all the DNA is and that's where transcription is happening. So yeah, that's basically transcription for you. So now we're going to move on to translation. So where does translation happen in the cell? Yeah, so it's going to move into the cytoplasm, it's going to exit the nucleus, and it's going to go to a ribosome, okay? And I'm just going to draw a simplified version of a ribosome for you here. All right. Okay, so now we're up to translation. Our mRNA strand, which was based off this DNA over here, has exited the nucleus and has gone to a ribosome. And it's inserted itself inside this ribosome, okay? Now, how is this ribosome going to read this mRNA strand? Does anyone know? In triplets. Yeah, in triplets. And what are the name of those triplets? Yeah, codons. So that's the first principle you need to know about translation, okay? It's not going to read it strand by strand, it's going to read it codon by codon. Okay? Now, what's another important structure we need for tRNA? So mRNA is one type of RNA and tRNA is another type. So if you've seen a tRNA molecule before, you'll know they look quite complex, but just for the purpose of this, I'll just draw it very simply. So this is a very simple structure of a tRNA molecule. Does anyone know what this top and this bottom bit of the tRNA molecule is called? Yeah. At the top we have an amino acid, and at the bottom anticodons, yes. So essentially what's going to happen in translation is we have our mRNA strand, okay, and it's divided up into codons. And what this ribosome is going to do is it's going to move along this mRNA strand. It's going to read the mRNA strand codon by codon. And what's going to happen is a tRNA molecule is going to enter the ribosome, leave an amino acid, and it's going to exit the ribosome. So how do we know which tRNA molecule and which amino acid actually enters the ribosome? Can anyone explain that to me? Yeah. So the codon actually determines which amino acid and which tRNA molecule actually enters the cell. So let's say our codon here is ACU. What's going to be our anticodon? UGA. Okay, remember this is RNA, so there's no T. Okay, it's only U. So we're going to have a UGA anticodon. Okay, on our tRNA, and that specific anticodon tRNA is also going to hold a specific amino acid. Now, does anyone know how many amino acids there are? There's 20 amino acids. And how many combinations of codons are there? 64. Okay, so that means we're going to have some codons which will have the same amino acids. Okay, that's just a small little fact you should remember. So yeah, as the ribosome reads the mRNA strand, there's going to be more amino acids added from more tRNA, and we're going to get ourselves a, what's this called? Yeah, it's a polypeptide, okay? We're going to end up with a polypeptide. And what is this polypeptide linked with? Yeah, peptide bonds, all right? You guys know everything, basically, so yeah, good job. You taught them well. Yeah. So that's basically protein synthesis for you. So this is a polypeptide, and what's going to happen, it's going to fold and modify to form functional proteins. You guys know about like the quaternary, tertiary proteins? You don't need to know them specifically, you just need to know that the polypeptide will fold itself to make functional proteins. So yeah, you guys know the process pretty well. Sorry for this messy diagram, but yeah. Yeah.
Okay, thank you. Yeah. Thanks, guys. I didn't even get to any information, so thank you. All right. What I will do, how are we going for time? I've only got 5 more minutes. Okay. What I want you guys to do now is go back to our notes, and I want you to have a quick read of electrophoresis and PCR. Because what I want to do, this is a bit ambitious, in the last 5 minutes, I want us to formulate an exam template answer for PCR, an exam template answer describing electrophoresis. Okay? So I'm going to pull this down. So we're about here. So have a read, and I want to see who can absorb this information and articulate it into an exam-style answer. The question is very simple. Describe PCR, describe electrophoresis. Okay? If you give me more than 3 lines, I will cut you off. Okay? Good.
PCR does. Shut up, I'm waiting. Double the quantity of DNA. And then RNA kinase is what? 93 degrees, is it that? No. Oh, it's not. So like, when you don't write it, it's like, so how does it really get this? Does he want us to speak to?
No, I think so. You can't ask.
Yes.
Okay, so you're doing very well. So I'm very happy with that. I never get a standing ovation teaching, so I'm quite disappointed. All right. So polymerase chain reaction. If you were to describe it, let's go with. Describe it. PCR. In the best of your ability.
Okay, PCR involves receiving a DNA.
I don't want you to read off the screen. I want you to read. Give it a go.
Okay, PCR testing involves receiving a DNA sample and amplifying the DNA through either manipulating the temperature and using a primer to further amplify the DNA and increase the weight if the sample has.
Pause. Okay, you did very well at the beginning,
Yeah.
but then when you start using thought, like almost like thought experiments, if the sample is like this by weight, then you go into a lot of convoluted information, right? Give it a go.
I've seen the gel ones, I haven't actually written them.
Okay, do the gel electrophoresis one. The game. No, looking down.
Okay, enables us to visualize and amplify DNA to specify whether the DNA sample has the disease.
Okay, good. That was a good outline. And all right, Prithvi. PCR.
First of all, PCR test is to amplify the DNA strands in a given sample, and this is done by breaking the hydrogen bonds in the DNA by changing temperatures and adding samples of the specific disease strain to check if there's the specific.
Run PCR.
I just said it was an artificial method of synthesizing nucleotide.
That's a define, but describe you want a bit more characteristics. Because what is describe, everyone? It's the characteristics of the process, right? One more person, and what we might do today is we'll probably end it at describing PCR. I'll explain to you my breakdown description. Mrna, what's yours for PCR? That's okay, but do you guys all see the hard part about biology? This is actually not really to teach you PCR. Is to show you that biology is two things. It's number one, understanding a concept, which is a whole game, and then learning how to write it down in an exam setting, which is a whole different game. The smartest students who understand the most in biology don't necessarily do the best because they can't articulate themselves well. You need to find that balance where you know the right amount and you can articulate things in a very concise manner. The thing I liked about every description is you all said it amplifies DNA, right? So that's the way you should start. PCR is a method of amplifying DNA. Now, what are the two things we use in PCR? We use ingredients, your enzymes, and we use a thermal cycler. So I've mentioned that. PCR is a process of amplifying DNA using, what do we use? Question? PCR, what does it involve? Yeah? Yeah, okay, so PCR is a process of amplifying DNA using a solution containing a primer and polymerase in a thermal cycle. Then the second sentence should be, what are some characteristics of that process? Cyclical temperatures of 55, 95, 55, and 72 are used to denature DNA and replicate and amplify the strands. Okay, so I have to very quickly write that for you all. PCR amplifies DNA using, mention the ingredients in thermal cycler. And the final thing you want to hear is the temperature changes, which is 95, 55, and 72 to replicate DNA. 2 lines. That's all you need for any description process. So remember this again. If it's a described question, that can be 1 line. That is the definition. Okay? And remember to state characteristics. Question?
2 or 3 lines.
Yeah. 2 would be ideal because you'd be very concise. 3 is fine. More than 3, you're going into more of a discuss or an explain question. Okay? Good. You are all look when you had a question before we finish up.
Yep. In the ingredients, does it contain free-floating nucleotides too?
Yes, that is another very important point. You are completely right. Yeah, we will talk about that next lesson because I did rush this. But next lesson I'll show you how to write the exam explanation for PCR.
Yep.
Okay? All right. Take care, everyone. See you.
Thank you. Have a good day.